Malaria is a state of disease attributed to 6 pathogenic varieties of a single-celled parasite in the genus Plasmodium. Of the 6 varieties, Plasmodium falciparum causes the most fatalities, particularly among small children and pregnant women living in sub-Saharan Africa. Malaria is older than mankind. It is a masterpiece of pathogenic evolution. Its life cycle is nurtured between a human host and a mosquito vector. Infection begins with a juvenile, motile form of the P. falciparum parasite, which spills from the insect's salivary secretions into the capillaries of the dermis, as the infected Anopheles mosquito takes a blood meal in the night. This begins the exoerythrocytic stage of malaria. The motile form is called a sporozoite. Now, it must clear the dermis, enter the systemic circulation, and reach the liver cells rapidly. But you see, crossing impeding cellular barriers is akin to traversing a no-man's land. Foreign molecular patterns on the cell surface might alert sentinel macrophages to engulf and degrade the invader. To this end, sequence homologies of parasite and host cell proteins quiet an immune response which might have benefited the host, had it not assumed pathogen proteins to be self. Perforin-like protein 1 is analogous to the pore-forming molecules secreted by CD8-positive T and natural killer cells. To cross host tissues, sporozoites will use vacuole shelters of endothelial cell lipid bilayer origin until pathogen recognition receptors in these membranes stimulate joining of lysosomal vacuoles to the parasite's shelter. At the slightest pH drops in the media, influenced by lytic enzymes, sporozoites will secrete the perforin-like protein 1 to polymerize, form pores in the membrane, and afford the sporozoite a rapid escape. In this fashion, an hour won't go by before the sporozoite has gained the sinusoidal cavity and established itself in a hepatic cell. Thinking back to inoculation, a portion of the parasites also entered the lymphatic circulation. A dendritic cell might have destroyed the pathogen and presented its polypeptide fragments on MHC class I or class II molecules to activate CD8 and CD4 positive T cells. By the time proliferation and evacuation to the periphery has occurred, it is likely that those antigens which the T cell is in search of are seldom found. The parasite has already invaded a Kupffer cell by means of its circumsporozoite surface protein. It has suppressed binding of NF-kappa B transcription factors to the DNA of that cell and upregulated IL-10 anti-inflammatory cytokine expression, ensuring safe passage to hepatocytes. It is known that the hepatic stage of development involves severely reduced pro-inflammatory IL-12 and TNF-alpha cytokine levels, but scientists are at a loss to explain the minimal MHC class I antigen presentation capacity of hepatic cells. It is suspected that in creating shelters of parasitiferous vacuoles, sporozoites hijack PRR signal transduction such that the vacuole remains relatively undisturbed. Incubation of one sporozoite in an immunosuppressed hepatic niche can create 30,000 merozoites capable of invading erythrocytes in the following clinical stage of disease. An asymptomatic exoerythrocytic stage implies controlled cytokine output. The erythrocyte stage begins just as mildly. Merozoites are surrounded with hepatocyte-derived membranes called merosomes, which mask their PAMPs. In minutes, the merosomes bind red blood cells and begin the blood infection stage. In the first cycle, merozoites mature into trophozoites and then divide, 1 cell to 16 cells. The first of these divisions influences the nature of PFEMP1 protein. This Plasmodium falciparum erythrocyte membrane protein is the product of one of hundreds of VAR genes which are distributed on 14 parasite chromosomes. Consider the mechanics we employ to extravasate leukocytes to sites of infection. The upregulation of cytokines TNF-alpha and interleukin-1 stimulates the vascular endothelium to express ICAM and VCAM ligand molecules. Now let's put this in the context of a macrophage, recognizing an infected red blood cell by way of antibodies having previously bound PFEMP1. This macrophage would secrete TNF-alpha, which in turn leads the vascular endothelium to express more ligands. The PFEMP1 of parasitized red blood cells bind with great affinity to these ligands. Such binding effectively removes trophozoites with the red blood cell from systemic circulation, helping them avoid clearance by the spleen. This mechanism confers a timely protection. Attachment needs only to be long enough for developmental completion so that the trophozoites are released into the extracellular space, ready to invade a new cell. Maturation cycles last 48 hours. Bursting erythrocytes release ATP, which is a damage-associated molecular pattern recognized by knot-like receptors of blood macrophages. NLR stimulation triggers formation of the inflammasome and caspase activation, cleaving pro-interleukin-1 and interleukin-18 to active secretory forms. IL-1 communicates with the hypothalamus, inducing pyrexia, and with the endothelial cells to express yet more binding ligands. Parasitized red blood cells can aggregate in deep tissue capillaries with other red blood cells in a process called rosetting. This often leads to ischemic organ damage, metabolic acidosis by respiratory distress, and overall systemic shock. Interleukin-1 and TNF-alpha cytokine storms every 48 hours create a picture called a tertian fever. Because free blood merozoites are such a fleeting existence, it is increasingly challenging for B cells to mount proper antibody responses against them. Antibody responses are more often mounted against the PFEMP1s, but remember that only one variety of PFEMP1 is expressed per cell, and that there are hundreds of repeat regions encoding other varieties of the protein on 14 chromosomes of VAR genes. Every 48 hours, 1 in 500 red blood cells will see a switch in the VAR gene expressed, presenting the repertoire of B-cell receptors with an entirely new antigenic sequence. Couple this with the erythrocyte's incapacity to present antigen on MHC molecules, limiting CD8-positive T-cell response, and you have successfully renewed immune evasion every 2 days. The blood stage of infection is still largely mediated by CD4-positive T-cells. As the parasite takes over, infected erythrocytes greatly outnumber dendritic cells, such that these dendritic cells experience antigen exhaustion. They meekly activate CD4-positive T cells, which differentiate into Th1 helper cells. Th1 is known for interferon gamma release, which activates macrophages to engulf yet more parasitized erythrocytes. By now, many macrophages that have done so are non-functional. When trophozoites are dividing, they metabolize hemoglobin for protein into an insoluble aggregate called hemozoin. This is extremely toxic to our immune cells. Hemozoin is long-lived in macrophages and deleterious to their capacity of MHC presentation and cytokine secretion. Some trophozoites will differentiate into gamete forms. B-cell defenses, antibody release, and opsonization mediated by these antibodies is most effective towards these free forms. But by this time, another female Anopheles mosquito has likely taken a blood meal from the infected host and furnished a safe immunosuppressed niche for sexual reproduction, genetic recombination, and sporozoid formation ready to infect a new human being.